Tim, who will be delivering on heredity. Now, just a reminder, you can post questions in the chat during this session, and Tim will answer those questions as you go along, unlike when we watch the recorded session. So thank you, Tim.
Thank you very much, Marnie. Yes, yeah, so welcome everyone. Yeah, my name's Tim Sloan, and I always get excited to step away from my own classroom and deliver, I guess, this annual event for me via Aurora College to you guys. My background, I grew up country as well, actually, so I love touching base with you guys as well. So yeah, look, it's interesting. It's a challenge from my end on how do you fit in a 2 or 2 1-hour sessions, approximately 20 weeks worth of work. So the way I'm going to deliver it is just, I guess, a mix of, I guess, key tips in terms of what I've taken away from my experience with HSC marking and judging, and some of the key focuses that sort of repeat themselves through each of the HSC exams, and then certainly have some HSC questions sprinkled throughout the presentation. Now, there is inevitably going to be some crossover between mine and the other presenters, or Sylvia's first session on how to move up the bands, but just again, some of these important pieces of information that I've, I guess, taken with me on my journey in as a biology teacher. You'll obviously be familiar with the glossary of terms, but just for some background information, a colleague of mine many years ago did a bit of analysis on several of the HSC biology papers, and it was sitting at around 60–70% of the paper really is pitched at that explain level. Okay. Now, the reason that I tell you that is that it's quite a common way where students do themselves a disservice and lose what are relatively easy marks, because they stop short and their responses tend to be at that identify and describe level. So it's really important that you understand what it means to explain. And look, the nested definition to me is a bit wordy, but as we move through the presentation, I'll draw your attention to some, for those that are probably better apt at English than myself, some of the conjunctions that will really sort of put you in a good place to answer some of these explain questions. Now, again, Sylvia's presentation delved a lot into moving up the bands. I just wanted to draw your attention to the band 6 descriptors, or performance band descriptors here. Now, obviously, as a teacher and for the students, the most important document is the syllabus, but second to that, I certainly believe the performance band descriptors are. You know, and the reason for that is that the band 6 descriptor here, I'm just going to draw your attention to a few of the key points, and I'd ask you just to answer the question yourself. Could you easily distinguish between accuracy, reliability and validity, because that's really is an expectation of a band 6 student, and that is both for primary and secondary data. Okay, so ensuring can you clearly articulate the difference between those terms, and a sure sign that a student is not performing at that band 6 level is where they go in their definition of validity, go and use that term reliability or accuracy in defining validity. We really need to be able to tease those terms apart. Now, the other one that I'm on a bit of a bugbear at the moment in my own class here is trends, pattern and relationships. It appears throughout both the Year 11 and the Year 12 syllabus. And as with accuracy, reliability and validity, just ask yourself the question, can you clearly distinguish between each of those terms and actually put it into practice when you see quite typically a graph or a table where you have to extract information from the images or the model provided. Now, the other real key descriptor that I think really sort of stands out for a band 6 student is they're able to, at that bottom point there, apply knowledge and information to unfamiliar situations. Now, the reason I draw your attention to that as we move through some of the questions in this session, quite often we'll have students, and again, I guess anecdotally, my own experience, their first thought is when they see a question is that, in my case, sir didn't teach me that content. Now the reality is quite often it's a skills-based question that they can apply the knowledge that is being learnt in class to that particular context for the question that sits in front of them, because all the information needed will be in the stem of the question, and I'll certainly point that out as we move through the presentation. Again, just really quickly, there are 7 key skills, working scientifically skills, which I absolutely assure you are going to be assessed in the HSC. your school-based assessment would have been made up of will have been made up of 60%, excuse me, skills and 40% knowledge and understanding. So each of the modules has that one knowledge and understanding outcome associated with it. And as you move through each of the 4 modules in year 12, you'll be basically ticking off each of these skills. And while we tend not to be as explicit in, say, we're teaching today kids, we're going to be looking at processing and analysing data and information, it often is inherent when we're graphing and extracting information from tables and graphs. Now, the other, just a couple, again, more background information. So I'm not going to go into detail with this. It seems to be quite commonplace across schools and teaching, not just in science, but if anyone is interested in more detail, you can put it in the chat there, or basically email me, I guess, afterwards, and I'm more than happy to spend more time on this with you so that we can get into the content of Module 5. And the idea behind this is that it's called the ALARM scaffold, but it's all about teaching students on that we learn in a chronological order. We learn in a specific order. Before you can explain something, you need to be able to identify it, describe it, explain it, and then move on to assessing or evaluating the key biology in our case. And so what we're trying to do is then teach you how to scaffold a response in a similar approach, where you really focus on a layered approach to your response, which I'll go through shortly with you on the next slide. And the idea is as follows: that if if you were to go home and ask someone who is not studying biology, ask your parents, your grandparents, could you evaluate the impact of a named transgenic species on society? Quite often their first question is going to be, what is a transgenic species? And so just highlighting there that before you can go through and hit those higher order verbs, explain and analyse, you really need to identify in the question I just posed what a transgenic species is. Once you understand what it is, you can go on to give some of the characteristics and features of it, explain the importance of that using your conjunctions. And conjunctions, I still use it in my own classes, joining words. So things we're looking for is like, as a result of this, this leads to, because of this. There's some really key, I guess, terms or words to have in your memory bank as you hit the exams. And then finally, again, a new acronym. Again, for me, we've been using it a lot in the last year or two. It's starting to be whole school. And this is just basically really encouraging you to hit the pause button before you start writing your responses in your upcoming trials and HSC exam next term. And it's, again, another easy to remember acronym. Put the pen down and just analyze the question. Circle the key verb of the question. Underline what it is that you're going to, or the key biology of the question. Any important plurals, and you'll see that's a big focus of my presentation today. The plurals are absolutely essential. So when you've got examples, that little S on the end of example is essential in terms of accessing higher marks. Okay, you've got 180 minutes to do a 100-mark question, and that equates to around 1.8 minutes per mark. And some of the multiple choice you'll be able to get out of the way, in 15, 20, 30 seconds. And so for those longer response questions, you really can be looking at 2 minutes per mark, and a significant amount of that, I strongly urge you to focus on planning the response and having that, applying that scaffolded response. So into our bread and butter, Module 5, heredity, the first inquiry question. And just to hit pause for a moment, and in session 1 up until 10:45, I will be focused on inquiry questions 1 through to 4. I personally find that Module 5 is a bigger module of work than Module 6. And then in the second session, I will look at inquiry question 5 of Module 5, and then the remainder, all of Module 6, just as that gives some background information on that. Now, the first of the dot points, okay, all outcomes, or dot points as I call them, are absolutely massive these days as compared to the previous syllabus. And just highlighting some of the key the verbs there, again, just highlighting you need to know what it means to explain, okay, and the different mechanisms of reproduction. And again, just delving into the outcome, you need to understand what it means to analyze data and information. We've got sexual and asexual reproduction, so you really need to be able to clearly articulate the difference between those two modes of reproduction. And the all-important, I was going to say all teachers, but so I'll say myself, I'm not a big fan of the including but not limited to dot points, because of what it means for us and you, the students. You need to know all of the examples provided in detail, and you also have to have some other examples up your sleeve. Now, quite typically in the HSC exam, how that would appear is via a STEM, or basically introduction to the question, that gives you a scenario and you need to extract information from that STEM. Rarely would they ask you to, with reference to example not studied in the HSC course, provide information on another type of asexual reproduction. It would typically be associated with that stimulus. And in terms of breaking down that outcome, so just at the top there, just remember those key conjunctions. So as a result of sexual reproduction in animals or plants, again, just making sure that you give that description of sexual reproduction as it applies, and then lead on from that to highlight the importance of it. Okay, you need to be able to articulate the difference between those key terms in the dot point. And I always find, interestingly, students can quite typically distinguish between asexual and sexual reproduction. But interestingly, when it comes to internal and external fertilization, often they're mixing up external fertilization and confusing it with asexual reproduction. It's really important. I'll just jump to the next slide and tell you why. It's really important that you understand that external and internal fertilization both relate to sexual reproduction. Okay. You really need to know how all these different terms apply to each of those different groups of organisms, the plants, fungi, bacteria, protists, and animals, and understand the advantages and the disadvantages of each of the terms. And some of them are just highlighted around this particular slide. With asexual reproduction, sorry, hitting pause again, it's also the reason why a lot of organisms are able to both asexually and sexually reproduce. They're basically taking advantage of what each of those different reproductive mechanisms offers to the species. And to highlight that, with asexual reproduction, there we've got what the downside of it is that they're genetically identical to the parent, which puts them typically at risk of, extinction or wiped out within a particular area if environmental conditions change. But so they're typically found in quite stable environments where conditions aren't changing. And then jumping over to sexual reproduction, the significant advantage of sexual reproduction is that it increases the genetic variation within the population, and therefore they're typically at reduced risk of extinction, or within a particular area being wiped out if environmental conditions do change. Then jumping down to internal external fertilization, this has certainly been assessed quite recently in the HSC exam, knowing the advantages and disadvantages of each of those. With internal fertilization, we're certainly more likely to result in pregnancy, and that's simply just due to that introduction of sperm and egg in much closer proximity to one another, or relative proximity to one another. There's typically more chance of survival of those offspring, simply because there's often, but not always, parental investment in the raising of those offspring. And arguably the most important, it's allowed life to exist on land, sort of sexually reproducing organisms, because if we were to rely upon external fertilization on terrestrial or land-based environments, the egg and sperm are at risk of drying out. The scientific word for that is desiccation. Okay, and so I'm just going to run through like this, and then we'll tackle some questions together. Analysing the features of fertilisation, implantation, and hormonal control of pregnancy and birth in mammals. The key tip that I'd ask that you take away from this, and again, I'll hopefully prove it to you when we look at the questions, is the focus is on that analyse. So we need to be able to extract information from graphs or tables, and then make inferences from it. Note though, we tend to focus, myself included in class, we do go through around 4 primary hormones that we ask that students have an understanding of how each of those hormones plays a role in pregnancy and birth. But what I'd highlight here is that no specific hormone is actually mentioned by name, okay? And so when the questions do come up, again, often associated with a STEM or stimulus that has the relevant necessary information contained within it, and alternatively, the question will be quite open where you have, I guess, open slather in talking about a hormone of your choice and how it impacts on pregnancy and birth. Okay, so again, just to reiterate, what does it mean to analyze? Again, because where students often lose marks is they stop short and, you know, they'll just say that a graph goes up or the hormone goes up or down or something to that effect. You really need to delve into the data points, identify specific components on those hormonal graphs and understand the relationship between them. So I guess in that sense, it's a causation, explain, and then draw out and relate the implications of that, which is the NESA definition of the analyze. So the dot point is very specific, fertilization and implantation. So you don't know which one or both, perhaps, that they're going to assess in the HSC. And bold there, hormone control of pregnancy and birth. So you need to be able to apply it to both pregnancy and birth. Okay, I've covered that. And again, just highlighting the two hormones I would best suggest that you focus on, and it will become apparent why on the next slide, is FSH and progesterone. For mine, they're just the easier of the four to look at and understand the role that they're playing in pregnancy in particular, and we'll look at birth after that. Here we've got, I guess, a pretty standard image. You need to know, I guess, the reasonable details of this whole process from fertilization, which is occurring down here in the fallopian tube, and then some of the key terms. As it moves along the fallopian tube, the zygote is the first diploid cell of the new organism. The other terms certainly are morula. Again, just highlighting, they're not explicitly mentioned in the syllabus, but I'd suggest that Band 6 students, those higher end students, are able to use that science meta language, which are those key terms that are associated in this case with pregnancy and birth. So the morula is just that collection of cells that hasn't undergone any specialization yet, and shortly thereafter, I'd strongly urge you to remember the term blastocyst, and it's the blastocyst which has started to undergo specialization. So we're getting some distinct cell masses that will go on, I guess, different lineages in terms of what they develop into, and it's the blastocyst that then implants down into the endometrium of the uterus there. Now, again, not that I'm writing the HSC questions. If I was writing them, quite often it'd present with a data or a graph like this. And I mentioned the two hormones that I'd suggested the bit easier ones to remember is follicle stimulating hormone. And the reason I say that is it's all in the name. What is the role of the follicle stimulating? Well, no surprises, it's to stimulate the release of an egg from the follicle in the ovary, okay? And then understanding that, it's almost, I think it's common sense that follows on from that, that you would expect a clear peak in that, or spike in, sorry, it's the green one here, in the follicle stimulating hormone. And once its job's done, so it peaks or spikes, the follicle releases that egg, and then the levels of that follicle stimulating hormone will then drop off and remain low until that process is repeated in the next cycle. And then I also mentioned progesterone. The reason I like this one is that it's heavily involved in initially causing the thickening of the endometrium in preparation for the implantation of the blastocyst. Now, one of the questions I will look at with you explores this idea with a stimulus where what would you expect if a female was to fall pregnant? What would happen to those progesterone levels? And as you look at that cycle, progesterone's involved in the thickening of the endometrium for in readiness for implantation, and as a result, if implantation occurs, fertile and pregnancy is successful, those progesterone levels would then stay high for the remainder of the pregnancy until birthing. And the reason for that is we need to keep that endometrium nice and thick to sustain the placenta and developing fetus. Okay. Now, again, I'm not saying you can't remember the other hormones, but they're the ones that I tend to focus my own teachings on. Now, this particular outcome is already falling behind time, so I'm going to have to speed it up. All right, I've actually changed my own delivery of this particular outcome in the last year or so, and I'll tell you why. Evaluate, so it requires looking at the positives, the negatives, but also making a value judgment. Again, don't do yourself short. An evaluate or assess really does require that value judgment. As teachers, we really value your input. Okay, you're the future. And so it's really important to have an understanding of what these different technologies offer, the pros, the cons, and then give an opinion. But in this particular dot point, what I want to draw your attention to is historically, I used to spend a lot of time looking at the specific technologies, looking at the pros and the cons of the technologies. But in recent times, as we review our teaching programs, you'll note that what you're actually evaluating in this case is the impact of scientific knowledge on the manipulation of these technologies. So our time spent on the reproductive technologies is not wasted because we look at them again in Module 6 in quite a lot of detail. But what you need to do with this particular dot point is have an understanding of the contributions of, I guess, a handful of scientists and understand how their work has contributed to the work of scientists that has followed on after them, and then how that's then assisted us in the development of these reproductive technologies. One of the quotes, and in essence, it's a brilliant quote. I'd urge you to remember, but just remembering this quote will help you to answer some of these questions if you have that background information. The concept is that the work of scientists, they don't work in isolation, and it's the accumulation of scientific knowledge over the course of time that has actually led to the reproductive technologies that are now quite widely used in many cases. I've largely just that through the middle there. I won't spend too much time on that, just to highlight the point that it's all about the scientific knowledge, that some that you might want to tuck away that you would have learned even in junior years is, and certainly in year 11, 12 content, the laws of hereditary. Gregor Mendel, he had no idea about obviously genes and alleles, but what he did bring to the table was that understanding of these factors that could be passed on or were passed on to subsequent generations. Now, armed with that knowledge, a whole lot of research went in between that, but we've got obviously Watson and Crick discovered the structure of DNA molecule, and from that we then went on to discover that DNA is common to all life forms, which then led to the development of recombinant DNA technologies where we can produce transgenic species. And more recently, all the rage at the moment is CRISPR-Cas9, which certainly has made an appearance in recent HSC questions as well. So again, just understanding how these developments don't occur in isolation, that is that incremental impact of scientific knowledge that has really come to the forefront in the development of these reproductive technologies. Okay, so having said that, I'll dabble a little bit with these technologies in Module 5 and in Module 6. Artificial pollination, and another point I would make is the outcomes, biotechs versus genetic technologies. It's really important that you do understand the difference between them. And in short, all genetic technologies are biotechnologies, but not all biotechnologies are genetic technologies. This is an example of a biotechnology. It's not a genetic technology because we're not actually manipulating the genes of the organisms. Hopefully that was clear, but again, throw it in the chat if that wasn't, and I will have it in an upcoming slide as well. So the key takeaway from artificial pollination is that it's not a guarantee that we're going to pass on those favorable traits to the offspring, but it certainly increases the likelihood. The downside of a lot of, but not all of these technologies is that then as a result of, I guess, choosing the parent stock is you're actually going to reduce the genetic variation or gene pool within the population, which therefore places them at risk, susceptibility of environmental changes or disease. Okay, just quickly, cloning. Again, it's applicable both gene cloning and whole organism cloning, which is what we're looking at here. That process involves producing genetically, the advantages are we produce genetically identical offspring to the parent of interest. The distinct disadvantages of that is that it very much reduces the gene pool, but also places them at great risk of, I guess, disease, environmental changes if we don't introduce variation through other mechanisms. Reasonable assumption that you understand the basics of cloning, but in short, with animal cloning, typically, but not always, we take a somatic cell, aka body cell, which is diploid, remove the nucleus, and we then insert that nucleus into the denucleated egg of another female. So denucleated, the nucleus, haploid nucleus is removed from the egg. That diploid nucleus is inserted into that now denucleated egg. That's then inserted into a surrogate mother, and then that surrogate mother will give birth to individuals that are identical to individual one. The all-important transgenic species, and so just to understand the advantages and disadvantages of that. The principle is that, and again, understand the scientific knowledge that has led to this point. This would not be possible if we did not have an understanding of the structure of DNA and understand that DNA is common to all life. Armed with that knowledge, we can now extract genes of interest from organism 1 and insert them into organism 2 so that they then express that gene from organism 1 in their phenotype. This is just what we call a novel approach to it, this image, just to highlight how the technology works. In this case, we've got a gene from a jellyfish. It's inserted into a recombinant plasmid, and that is then that plasmid is then inserted very early on, quite typically the egg sperm fertilization stage, with the hope that that transgene is actually inserted into the resulting offspring, and then those offspring will be able to express that gene in the form of a protein in their phenotype. Okay. Oh, sorry, just reading the chats there for a moment. All right, so just to put that into practice now, this is, I've tried to make them as current as possible, grabbing questions from the 2021 through to 2019 paper as well. So which of the following photographs shows an example of sexual reproduction? Okay. And this is just to highlight that point that I find quite often students actually mix up the external fertilization with asexual reproduction. So I'm not sure whether it's a mix of teachers and or students on the line, but does anyone want to throw in an answer there in the chat, or if nothing appears, I'll just go through it and explain why. Nothing's coming up. The answer is, okay, sexual reproduction, it is going to be D. Okay, frog spawning, that's external fertilization. And just yeah, really highlighting that in this particular image, they've covered 4 groups of organisms from that particular dot point that we looked at the start of the presentation. So you need to be able to apply your knowledge base across all of the different groups of organisms. Again, a multiple choice that's just tapping into your understanding of fertilization implantation. Whilst the terms themselves aren't particularly problematic, it's slightly tricky, these types of questions, in that you're having to juxtapose between the numbers and the letters at the bottom of the page. Obviously, well, hopefully it's obvious. The answer is going to be C, that ovulation, the release of the egg, first occurs. Once that occurs, the egg is fertilized. In mammals, it's in the fallopian tubes. We then get implantation and then followed by placental formation. As I said, that placental mammal, just another point I wanted to draw your attention to. The dot point is not specific to humans, it is to mammals. And what that means for you is that we tend to come at these dot points with a human focus, but again, don't be thrown if another mammal is provided to you in a stimulus. The same, I guess, knowledge base is going to apply in terms of how these hormones function. For example, we're dealing with in females, the menstrual cycle of approximately 28 days. with different types of mammals, that is going to be of a different length time. Okay, so don't be thrown. Just apply the knowledge that you've learnt with humans to any scenario that is thrown your way. And again, just highlighting here, you need to know the differences or the advantages of internal versus external fertilization. And with internal fertilization, it was one of those big evolutionary jumps that allowed organisms to go from water onto land, and relatively straightforward, I hope, in that it's going to prevent dehydration. Again, I don't think they would use dehydration. The sciencey word is desiccation, but it's all about ensuring optimal opportunity of survival for the resulting fetus. Okay. Interesting question. Certainly requires a good. This is probably across actually different dot points. This particular question, but certainly does apply in terms of having an understanding of sexual reproduction, diploid, haploid, but how that then applies when we look at it to meiosis. And quite often we don't use the term trick question as markers, but what we do use is distractors. Okay. So option D there is the distractor, because what students who don't have a really good understanding of meiosis will do is that they'll simply add 62, 44 together and come up with 106 and think they've got the correct answer. But you need to understand that it does tell you, just looking there, that this is having an understanding that meiosis is going to produce those haploid gametes. So in actual fact, you have to add 31 with 22 to come up with hopefully 53. So some of the shorter response one now, and this is just going to hopefully sell that message to you that the questions will not typically require you to, they're not going to ask you outline how progesterone by name changes over the course of pregnancy. They'll be often associated with these skills-based questions. And in this case, you can see that they've given you, it's the hormone is the human chorionic gonadotrophin, HCG, levels produced over the course of 40 weeks of pregnancy. Okay, so your initial thought might be, I haven't learned anything about this particular hormone. It doesn't matter, okay, because all the information you need is up there in the stimulus. And in this case, what they're doing is assessing your ability to graph that information. Okay, so in all cases, your teachers would have sold you this message as well. You need to know your variables. Your independent variable always goes, well, nearly always goes across the x-axis. Your dependent, the one you're measuring, is going up the y-axis. And other key tips are you must include units in your axes. And I'd strongly urge you to have include the title. You just don't know what the marking criteria might have, and that title doesn't need to be particularly fancy, but engage with the dependent and the independent variable. So in this case, it will be, how does HCG levels change over the course of pregnancy? Okay, and to highlight that, and then this is always a big focus of mine and will be for a lot of this talk, is looking at how the marking criteria is applied to particular questions. Okay, so this is a 3-mark question. The 3 out of 3 there is on the left. And just to highlight again, what we've got is the correct dependent variable with units, weeks of pregnancy across the x-axis, and note a nice title. And the other big thing that they can sometimes deduct marks for is if you don't utilize all the space provided. Okay, so you see in the graph on the right, which I had at a 2 out of 3. We've got all this wasted space up here. They've drawn a line of best fit. Sometimes that is the right thing to do. In this case, it certainly wasn't the right thing to do. So just, I guess, don't go, losing easy marks. And the other really important thing I will continue to draw your attention to is what you need to do for 1 mark. Okay, in nearly all questions, unless they're worth 1 mark, for you, if you were to provide some relevant information, you will score marks. Okay, so simply putting, in this case, concentration, HCG or weeks pregnancy on the axes without actually drawing the graph, I guarantee you would have got 1 out of 3. Okay, so if you apply that same logic across the entirety of your paper, it's just a surefire way to accumulate marks. So even if you're uncertain of the answer, rather than rushing leaving it out puts pen to paper. Okay, it's a really important point I'd like to make. Now, again, back to that earlier point, you can see here part B of that particular question. Describe the role and changes in the levels of a hormone in pregnancy. So that question is quite open. You've got the choice of hormones that you want to address. And again, to reiterate, progesterone or FSH is the one that I would certainly go to, and the choice is yours. If you want to go for one of the other more complex ones, absolutely do. Just note here, there's the question, and we'll do so for the remainder of the talk. Just highlight how the question actually marries up with the marking criteria, okay? You were required to, again, analyze the question, cube it, circle the verb. It was a describe, okay? So note for 3 marks, what did you need to do? No surprises, describe. Now, the other key points within this stimulus is the role and changes, okay? Quite often students will give a really good description of the role of progesterone, but they didn't go on there, or if that was your choice, but they didn't go and look at how the levels changed over the course of pregnancy. And again, you're instantly going to be dropping marks simply by ignoring the and. Okay, and highlighting a hormone, your choice, and it's to do with pregnancy. Again, it seems obvious to me, but apparently not in the student world. Quite often it wouldn't be unheard of that they would go and talk about oxytocin, okay? Oxytocin, again, I think it's a good example, more so involved in birthing, and so students will know a hormone of each, but because they haven't spent the time analysing the question, what then happens is that they will still access marks for sure, but this particular question is specific to pregnancy. So your year, this year, in a few weeks or months' time, you might get a question on pregnancy, on birth, or hormones related to birth. Don't go and talk about FSH or progesterone, because again, you're going to end up with 1 out of 3 if it was a similar mark question here. Okay. This particular question comes from last year's paper. It's actually not in the booklet, so it was a late call up just because I quite like the question. And it is just trying to make it larger for myself. So this is, again, a skills-based question that requires you to apply the knowledge that you've learnt in class. So note here, they give you all the information required, and it's referring to luteinizing hormone, okay? And this is ensures that students have an understanding of variables and experimental controls, more specific, or validity and experimental controls in this case. So it required students to, which of these results indicates a valid test, which shows that that ovulation is not occurring, and you had to then justify your response to that. And again, so if you can answer that in your mind, I'll give you 5 more seconds to have a look at that and ponder before we look at the marking criteria for it. Okay, and just as a reference for all the questions I look at, we'll have I put top right there, the stimulus, just as a reference point. And again, had we cubed the questions I refer to, which of the results, so you'll see here that lines up with the marking criteria, which is identifying, in essence, which of the results. It requires you to identify which is the valid result, and it is actually going to be strip, I better make sure I'm getting this right myself, strip 2. As I'm looking at it, it's a bit small here. Yes, so strip 2 is the only valid negative result. And note, the question required you to justify your response. So what that then means is that you then need to engage with the actual stimulus and talk about the different lines that appear on each of the strips, okay? So if you didn't do that justification, you'll see here that you're instantly going to be down at 1 mark, because even for 2 marks, you needed to provide a justification, okay? And so in this case, that justification, the control line is present in sample 2. No LH surge line has been detected in strip 2, and therefore it is a negative test, okay? Just getting a sense of where I should be up to, okay? Moving on to inquiry question 2, and it's all about DNA structure and the processes of mitosis and meiosis. Again, we've got the including but not limited to, my least favorite wording within the current syllabus. And I've grouped, for reasons that will hopefully be obvious, the next dot point 12.2b, which is looking at the effect of cell replication, be it mitosis or meiosis, on the continuity of a species. Models take all different, I guess, appearances. They can be computer models, an image, just a simple diagram. You drawing a flow chart would be you demonstrating your ability to model mitosis and meiosis, okay? It's essential that you understand that complementary nature of DNA that was discovered by Watson and Crick, and the importance of that in terms of DNA replication and subsequent protein synthesis. Yeah, look, I've said I'm willing to bet my career on it that you will be assessed on meiosis and mitosis, probably, well, both, but more so meiosis. And the reason I say that, it is just one of those absolutely fundamental concepts within biology where you're required to understand how the different stages of meiosis introduce variation into the species. And it is that genetic variation that is responsible for survival of a species, but also ongoing evolution and speciation. Now, down here, some tips I've learned along the way. If asked to draw a flow chart that demonstrates different stages of meiosis or mitosis, that word flow chart means that you must include arrows. It gives direction, flow, okay? I've seen students that give amazing diagrams. If I jump forward to images of meiosis, mitosis, you'll see this top one has arrows. A lot of really good information there, but had you left the arrowheads off that image, the markers would argue, I don't know which direction you were telling me the images are flowing, and you instantly start to lose marks, okay? Which might seem unfair, but a flow chart is a flow chart, okay? Just thinking, yeah, that shall, and give a value judgment. That's really important, because if you remember, it did require an assessment about the importance of both of the continuity of species, and both are absolutely essential, okay? Look, that image is readily available, but in the interest of time, I'm just going to focus on this one, the key differences. Parent cells, both diploid. With a mitosis, we've got those two daughter cells that are genetically identical. And the reason that's important for the continuity of a species is that it is those chromosomes that contain the genes that produce proteins that will code for proteins that are essential for the day-to-day existence of that, that organism. Now you compare that to meiosis, and I'm going to explore in more detail shortly the key steps: crossing over, independent assortment, random segregation. Okay, so what we've got here with meiosis is 4 daughter cells produced, and they are genetically different to that parent cell, okay? And that's absolutely essential to survival of the species, because this is where the variation is being introduced, okay? And what I want to look at on the next slide, it's taken me about 20 years of teaching to find an image or draw an image that sums up nicely the difference between random segregation and independent assortment, okay? Perhaps more importantly, just jumping back briefly to that last slide, crossing over it's an absolute essential to remembering, know the differences between homologous chromosomes and sister chromatids, okay? Quite often students mix those up, and they're sort of real fundamentals to understanding what's going on with meiosis, because it's the homologous chromosomes have two sister each homologous chromosome has 2 sister chromatids. So just trying to find one that I can draw your attention to here. At the top here, these 2 blue ones are 2 sister chromatids, but the blue and the red are homologous chromosomes, okay? So crossing over actually occurs between the sister chromatids of the 2 homologous chromosomes. Hopefully I've made that clear. But in terms of random segregation and independent assortment, this is the best way I can explain the difference to you, okay? With I'll jump to independent assortment first. Now, we've got roughly 25,000 genes spread across 23 pairs of chromosomes. Basically, the genes that are found on chromosome 1 will independently assort to the chromosome, sorry, the genes that are found on chromosome 2, that are found on chromosome 3, and so on. Okay, so at the top here we've got the parent genotype. We've got a gene A, but it's heterozygous. We've got one of each of the alleles, and then we've got another gene, in this case B, and you will note what A is doing in terms of which allele, the capital A or the little a, which one ends up in the daughter cells, is independent of what occurs to the alleles of gene B. Okay, so it's occurring independently of one another. Now, in contrast, random segregation is referring or relates to the one gene. Okay, you've got different alleles, in this case, capital A, little a, of that particular gene, and basically they will randomly assort themselves or segregate into the daughter cells. And so in this case, capital A is on the left, little a is on the right. It could equally have been little a on the left, capital A on the right. Okay, hopefully that has made sense. And I'm way behind schedule, so I'm going to skip through some of the questions, again, in interest of time. I'll just do one of these, the top one. I like these questions. It requires a little bit of thinking and having an understanding of the DNA structure. We've got 30% of DNA is adenine. What percentage is guanine? Okay, so what you need to appreciate is that the A is joined to the T, and therefore there is also 30% thymine, and combined that equals 60%, and therefore the guanine and cytosine makes up the difference, which is 40%, and therefore the answer is going to be A, 20% of the DNA sequence will be guanine. Just going to go through and choose a couple of standout questions for you. Again, 2014 question that does relate to these reproductive technologies. So scientists have tried to achieve a viable embryo fusing 2 ova eggs from the same female. Explain, there it is again, explain whether the offspring produced would be clones of the female, and use your knowledge of gamete formation and sexual reproduction to support your answer. Now, I'm not going to delve too much into the depth of this answer, but I wanted to draw your attention to a few things. Note, if anyone answered yes, that it would be a clone, then you're wrong, unfortunately. But I just wanted to highlight that if you did say that it was a clone, you're going to end up literally, no matter how good the rest of your response does, if you look at this marking criteria, you're going to end up with a maximum of around 2, maybe 3, depending upon the rest of your response. Because even for a 3 out of 5, appropriate assessment of whether the offspring produced is a clone. Okay, the answer is it's not. And hopefully you understand why. Is that those 2 eggs have been taken from the same mother? Yes. However, the production of those eggs through meiosis results in terms of crossing over, independent assortment, random segregation in genetically unique cells. Now, this is another really good question to highlight some key points. Again, it's a flow chart that was given to students, and you basically needed to fill in the gaps for 3 marks, like seemingly an easy question, but I'll tell you some of the pitfalls where students really messed up with this particular question. And I'll do that via actually the next slide. What students ended up doing is, you'll note that you had to follow the sequence and understand that the homologous chromosomes were actually lining up along the equator of the cell. And if you did not go and draw in this region here that I'm circling, if those sister chromatids weren't joined at the centromere, if you're rushed and you're not the neatest of drawers and you had a bit of a space between those sister chromatids, instantly lost 1 mark, because it demonstrates to the marker that you don't have an understanding of sister chromatids being joined before they then go and cross over. Now, the other important thing to note, it seems obvious to me, but as students did it really poorly, this is now all about random segregation. And once we've made that initial meiosis 1 split where we've got these 2 daughter cells, at that point, the results are fixed on each of those different sides, and these 2 daughter cells over here can only get combinations from this cell here. Okay, a lot of students felt that they were bringing over, oh, can't see it myself, they were bringing over the capital B over to the right-hand side, and that simply indicates to the markers that you don't understand that after meiosis 1 occurs, we've got those initial 2 daughter cells, and that you need to then follow that through to its completion and in the production of these 2 particular daughter cells. Okay. Now, I'm going to just skip because I'm a little behind time. It's important that you do know this image here, DNA structure. It's largely consistent between prokaryotes and eukaryotes. There are differences between them that you must be aware of. And some of the key ones are, as I think of them, the eukaryotes have those non-coding regions, the introns that are that are spread between the coding regions. And in order to get more DNA into the cells, they're wrapped around those histone proteins. Okay, the prokaryotes are lacking of those 2 particular non-coding regions and histone proteins largely. So that's if asked to compare them, a table is always great for a comparison. But once you understand the similarities between them, the information in regards to transcription, translation are the same. Okay, so don't be thrown if you get given a prokaryotic example. In terms of assessing the importance of mRNA and tRNA, I'd strongly urge you to fall on the side of assessing or giving a value judgment that they are absolutely essential. And the reason I say that is without them, you're not going to get correct protein production. Okay, and the all-important image, again, there's no easy ways about it. You will be assessed on protein synthesis and quite likely the impact of mutations on that process. So you really do need to know all the key players within this particular image that's shown here. Okay, so understand transcription. I always tell my kids to be wordsmiths, break the words down. Trans means to move, scribe means to write. So we're using the same language, in this case nucleotides, and we're moving it from DNA to mRNA, okay? That mRNA sequence then moves out to the ribosome, and it is that point that we translate, okay? So think about translating languages. We might be going from my school, Korean to English, English to Korean, and so we're actually moving the language from nucleotides to amino acids, okay? Absolutely fundamental area of biology that you must have quite a deep understanding of, and understand the impact that mutations have on that process. Okay, mutations only occur at the DNA level. They do not occur at the mRNA or the tRNA level. So don't be thrown if that scenario comes up, and it has certainly presented itself. Now, often if asked in a class, are mutations good, bad, or neutral? Most students typically fall on the side of bad because we sort of focus on, cancers and things like that. I understand why, but you need to also understand that mutations are also really positive. They're essential to the process of evolution, and quite often, or more often than not, they're going to be neutral if there's not a change in the actual amino acid sequence. And to highlight the bad side of things, we've got a normal strand of DNA. Whilst it's not explicit within the syllabus, you'll see discussions around template strand and coding strand. It's easy to get them confused. Interestingly, the mRNA that is formed, it is copied off the template strand. So it forms basically the template off which the mRNA forms. And so therefore we call the other strand the actual coding strand. So in this case, it's the one at the top is the coding strand. And you'll note that the sequence of that is identical to the mRNA, with the exception that thymine is replaced with uracil, okay? And I would suggest that you are definitely going to need to have an understanding of your codons and anticodons and be able to engage with those mRNA wheels where you actually are able to determine which amino acid would be coded for based on the codon present within the mRNA. Time-wise, we've got about 6 minutes left on this session. Okay, effective gene and environment on the phenotype. Look, this I find is the best example. Most of you are probably familiar with this. It is a turtle, but the crocodiles, saltwater crocodiles, are very similar. having an understanding of how genes influence, and environment influence the phenotypic expression. We've looked a lot at genes because it's the genes that contain that nucleotide sequence that produce the proteins, but environment actually influences how those genes are expressed. Best example I can come up with is here, where there's what is called a thermosensitive period. And in this case, between 28 and 52 days, the temperature at which the eggs are kept influences the expression of what's referred to as the SOX9 gene and dictates whether the offspring born will be male or female, where 25 degrees results in all male, all temperatures of 30 degrees are born female. So the warmer temperatures produce females, and I'd envisage that if it's somewhere between there or fluctuating, that you're going to get a mix of male and female. Okay. You do need to know the different levels of protein structure, that being primary, secondary, tertiary, and quaternary. The primary is just that specific sequence of amino acids. The way in which those amino acids then fold back on one another is what we refer to as the secondary structure. And then when all of those secondary structures then fold back and come together to form that overarching structure, it's referred to as the tertiary structure. Now, not all proteins have a quaternary structure. Okay, some proteins are made up of only one polypeptide. Others, like hemoglobin, I think it's hemoglobin here, it is, have what's referred to as the quaternary structure. Okay, that's made up of 4 subunits or 4 polypeptides, and what they do is then come together. Each of their tertiary structures come together to form this quaternary structure, which is the overall shape of these 4 polypeptides that form the final product. Okay, now I have 3-4 minutes left. This was a question that, again, 2014 question, arguably I liked it, but I also was empathetic to students because it was probably one of the hardest questions that I've seen come through. So if you can look at these, we spend a lot of our time talking about one gene, one protein being produced, and expect to remember band 6 students are able to apply learnt knowledge to unfamiliar contexts. So this particular question blows all of that one gene, one protein concept out of the water, because what we've got here is a specific reaction where, dictated by temperature along the x-axis there, the particular species of fish has 4 different genes that are making 4 different proteins in order to perform the same reaction. Okay, the number of students that got question 20 correct in particular, let's just say it's about 1 in 10 or less actually of students, of 18,000 students. That's the word that I've got, actually got question 20 correct. Think about which answer you're going to put in quickly. What does this case study demonstrate? If you've had time to read the stimulus. If you've locked in B, be proud of yourself. The reason that was quite complicated to get was the others are really good distractors, all 3 of them, but also in class as a teacher, myself included, up until 2014, we talked about phenotype as being the physical expression, what we observe, hair color, eye color. I certainly put my hand up and say that I wasn't going into enough detail to highlight that the production of a protein, enzymes, we can't physically see them with the naked eye. However, that is the phenotypic expression of that particular gene. Okay, so it's a really good question to highlight that last dot point that we looked at, which is influencing or looking at how environment actually affects the phenotype of an organism. Okay. We haven't got to all things Punnett squares. I'm not sure, Money, if you're there, would you like me to pause now for our break, or?
It's up to you, Tim, whatever you prefer to do.
Decisions. Thank you kindly. What I'll do then is I'll just intro this next dot point, because it's something that you can't rush through. Obviously, again, it is an area you are absolutely going to be assessed on. Okay. Again, the dreaded including but not limited to, you must know how to draw and how to interpret autosomal, sex linkage, codominance, incomplete dominance, and multiple alleles, and be able to construct and interpret both pedigrees and Punnett squares to optimize the marks that you get from them. So I think what we'll do, I might pause it there and then I'll hit fast forward next session in, 15 minutes time, where I'll just go through the basic theory of each of those different types of inheritance and look at some quite tricky at times Punnett squares and pedigrees. Okay, so
we'll pause it there. Thank you, Tim. Whew. That's why I put Wonder Woman running through the traffic, because you were just running at 1,000,000 miles a minute there. And I know that there were students madly writing as well, because somebody messaged me saying my students are madly writing as they're going along. So guys, take a half an hour break now. Please go and get a cuppa, stand up, walk away. I'll share the next link for the next meeting in the team. So I'll end this meeting and then we'll create another meeting so that we've got the recording separate. And I'll stop the recording now.